now in unit 1 we will start with the 1.4, that is concept of active site mapping, or you can say active site mapping. Until that I have told you that active site at which the substrate bind NES complex is formed. What is their role? How it is binding? What is the induced fit and lock and key model? How it is attached? Now concept of active site mapping. This enzyme refers to the identifying the process and characterize that specific region within the enzyme where the substrate and enzyme binds undergo catalysis reactions. So, as I said previously that the active site is a three-dimensional structures or a pockets or a cleft from the enzyme. That enzyme surface is complementary in the shape as well as in the chemical properties to the substrate molecules. It is within the active site that enzyme catalyze the specific chemical reaction by facilitating the formation or a breaking of chemical bonds. That you have to remember why it is facilitated. Now, active site mapping. Remember, what is the functions? How you will understand what is the application of active site mapping. So for that, active site mapping play a crucial role in understanding the enzyme functions, specific enzyme function. Because in living system you will find a series of reaction which enzyme one to another product it is formed. But that product is also an initial stage for another pathways. So that as a it provide an insight into molecular mechanism underlying the various enzyme reactions. So by identifying or characterizing the active sites, scientists can gain or deeper understand how the enzyme recognize and bind with the substrate, as well as how it was stabilized. So there are they stabilize transition state and another is facilitated the catalysis. So one of the primary techniques for the active site mapping is X-ray crystallography. So these X-ray crystallography, before applying that, you have to work on the substrate, enzyme substrate involving crystallization. And after the crystallization of enzyme substrate complex, you have to bombarding the X-rays. And the resulting diffraction pattern can be used to determine the precise arrangement of atoms. Precise arrangement of atom and the complex including the active site. By analyzing this structure, researcher can identify the key amino acid residues within the active site and that interact with the substrate. Now another technique that is employed in the active site mapping is site directed mutagenesis. So in site directed mutagenesis approach, they are introducing the specific mutations where in gene encoding the enzyme to alter amino acid residue within or near the active sites. Remember, in that approach, in site-directed mutagenesis, they are introducing the specific mutation at active sites. And that active site or near the active site, how these mutations affect the enzyme activity, as well as the substrate binding or a catalytic efficiency, scientists can interchange or infer which amino acids are critically for active site functions, or they confirm, or you can say they infer which amino acid is inferring for that particular reactions for determining the active site functions. Additionally, there are a computational method also there. This computational method, such as molecular docking, there are a large number of software you are available in the markets, as well as in the you can search in the Google also they will give you a URL for that. So molecular docking and molecular dynamic simulations are used to predict and explore potential interaction between enzyme and substrate. So, for example, molecular docking with the certain proteins. So, AMPA, antimicrobial peptide. Okay, AMPA is one of the software. You can put the sequences of amino acid or protein, and they will determine the how much protein or which peptide is responsible for antimicrobial property. Similarly, you can also use a molecular docking, various bioinformatics tools for determining this sort for predicting or used to predict and explore a potential interaction between enzyme and substrate. These techniques, this molecular decking and molecular simulation, or you can say molecular dynamic simulation, are predict or explore that generally it is relying on mathematical algorithms and a physical model to simulate the behavior of molecules. So in silico, there are a large number of papers you will find in silico mechanism of so and so enzyme substrate bindings of particular. So allowing researcher to predict how substrate might bind to the enzyme active site and how these binding affect the catalysis. I will also practically explain how to use that software for determining this molecular docking with the enzyme substrate or active site mapping, that I will also give you a practical. This is not in our course, but I will show you. Active site mapping has a numerous practical application in various fields. As I said, if you do not have anything, only you have a computer with the internet connection, you can write number of papers. The word will start with the in silico. In silico means you are not performing biological. You are utilizing a various software. Okay. So including drug discovery, biotechnology, and enzyme engineering. There are certain topics you can learn with this drug discovery, biotechnology, and enzyme engineering. This molecular docking as well as molecular dynamic simulation are used. And by understanding the active site of an enzyme, any scientist or you can design and develop a specific inhibitors that target the active sites. And if the enzyme is very toxic, then you can inhibit by this way by binding with the specific inhibitor to a specific active site or thereby modulating enzyme activity also. So one of the knowledge you can use for that also. And these knowledge also enable a rational design of enzyme with improved catalytic properties, improved catalytic process for the industrial application. So one of the use of these for various industrial application by changing uh, mechanism or active site mapping or drug discovery, or it is also used in the enzyme engineering. And nowadays in biotechnology, there are a large number of enzymes are this active site mapping is still in working conditions. So in nutshell, I can say that active site mapping is an enzyme or fundamental process that involving identifying and characterizing the specific region where the substrate bind undergo the catalysis. And for that, this active site mapping, you require X-ray crystallography as well as another technique that is site-directed mutagenesis. And there are two in silico model, that is computational modeling, commonly used to illustrate the structure and functions of the active site. And this knowledge is crucial for understanding the enzyme mechanism, designing inhibitors, and engineering the enzyme with the enhanced properties. So this is all about the active site mapping, and we will